Answering MCAT questions in under 60 seconds. Acetyl-CoA is both a precursor for fatty acid synthesis and a product of beta oxidation. Malonyl-CoA, which is involved in fatty acid synthesis, inhibits the transport of long-chain fatty acids into the mitochondria, preventing beta oxidation. If a drug is hypothesized to increase beta oxidation in cells, which of the following experimental results would provide evidence to support this hypothesis? Okay, so A says adding malonyl-CoA to drug-treated cells causes an increase in the level of cytosolic acetyl-CoA compared to untreated cells. Well, remember that cytosolic acetyl-CoA is going to be involved in fatty acid synthesis, right? Whereas acetyl-CoA is also involved in beta oxidation, but specifically it's the product of beta oxidation, so you would get mitochondrial acetyl-CoA, right? So having an increase in cytosolic acetyl-CoA is not really telling us anything about beta oxidation. In fact, if anything, it could probably imply less beta oxidation and more fatty acid synthesis, since those are sort of opposing processes. So A is wrong. B, adding malonyl-CoA to drug-treated cells causes a decrease in the level of mitochondrial acetyl-CoA. So that's wrong, right? Because mitochondrial acetyl-CoA is a product of fatty acid oxidation. And so if you're seeing a decrease in that, you're actually having a decrease, not an increase in beta oxidation. C, treating cells with the drug results in a decreased concentration of cytosolic malonyl-CoA. And that sounds like the right answer, because they're actually telling us, and we could know this too, that malonyl-CoA, it's inhibiting long-chain fatty acid transport. And that is a process that's required for those things to be oxidized. So if the drug results in a decreased concentration of cytosolic malonyl-CoA, you're disinhibiting long-chain fatty acid transport and therefore promoting beta oxidation. So C is good evidence, right? Whereas D, treating cells with the drug results in an increased concentration of cytosolic malonyl-CoA, that's saying the opposite. And that would actually be reflective of less beta oxidation, right? So C, therefore, is the right answer.